Hello, good evening, and welcome to the week 12 live session of Immunology. I am Surasree, and this will be our last live session for this session, for this run of the Immunology course today. And yes, so today is the last session, and in today's session, we will be discussing about vaccination and one very relevant technology, which is the production of monoclonal antibodies, which is the Hybridoma technology. So first we will talk about the immunization and vaccination. So what is immunization? Immunization is this process of eliciting a long-lived protective immunity against any disease-causing pathogen. Right? This immunization, this long-lived protective immunity, can result from various events. One is when, suppose, we are naturally infected by any pathogen or any microorganism, then what happens? And if our body can fight that pathogen, then after few days we start to show adaptive immune response, and our adaptive immune response also produces our memory B cells, which can remember this particular infection, and whenever there is a second round of infection, then these particular memory cells can act against the pathogen in a much better and more efficient way, right? Also, this immunization can result from various other artificial methods, like for example, one can be a passive immunization, that is the anti-serum. For example, in last session we were also discussing about this anti-venom. So whenever we have a snake bite or a bite of a poisonous snake, then what we do is we inject the patient with anti-venom, right? And anti-venom is raised, this anti-venom is basically raised in some organism like a horse or something. So that provides antibodies against the venom. So that is a passive immunization process, whereas vaccination is an active immunization process. So I'm going into more detail what is an active immunization, what is a passive immunization. But remember this, that vaccination is this intentional exposure to any forms of a pathogen, right? In form of a vaccine. And remember that vaccination is an event, whereas immunization is the potential outcome of that event. So why do we vaccinate? So that we can generate a long-lived protective immunity against that disease-causing pathogen, right? So that is immunization, that is the outcome, which is the immunization, right? Okay, next moving on to passive immunization and active immunization. So passive immunization is the process whereas the antibodies are transferred to a recipient, and it does not involve the activation of the B cells, because the preformed antibodies are being transferred to the particular recipient, which who requires this kind of antibody mediated protection and quick elimination of any toxin or quick elimination of any pathogen, right? So this passive immunization can be natural or artificial. What does it mean? So when a baby is in the mother's womb, then this passive immunization or passive immunity occurs naturally, because we know that the maternal IgG can cross the placental layer and it can come to the developing fetus, right? So the maternal antibodies come to the placenta and it immunizes the fetus passively, whereas after the birth of the baby also, the maternal antibodies which are present in the breast milk like IgA also provide passive immunity to the young baby. Right, so that is natural passive immunity. It is occurring naturally. We receive it naturally. But artificial passive immunity means that it can be achieved by injecting a recipient with preformed antibodies called antiserum, either from immune individuals, or we can produce these antibodies, we can generate these antibodies in individuals where this venom or the toxin is injected, right? So like I told you, for example, antivenom. Snake antivenom. So most important point here is that passive immunization does not activate the host's immune response. Why? Because the host is receiving these antibodies which can quickly neutralize the toxin or the venom or any other particular antigen. So therefore there is no need of the immune system to produce the B cells, and the humoral arm of the immunity is not activated, right? So the host immune system is not activated by this kind of passive immunization. Whereas in case of active immunization, we see that it triggers the adaptive immune response of the host, which elicits a protective immunity and an immunological memory also. So this active immunization can also occur naturally or artificially. How can it occur naturally? So if we are naturally infected by any pathogen or any microorganism, it can be achieved naturally. So like in the first time the body fights that particular infection, and then the subsequent infection elicits a secondary immune response which is much more quicker, which is much more efficient, and it successfully eliminates the pathogen and prevents the disease by mediated by its products. What are these products? Like, for example, antibodies, right? Or active immunization can also be achieved artificially by administration of a vaccine. So now we have come to this point where a vaccine is used, and vaccines are what? Vaccines are different forms of the pathogen which are injected in a healthy host, so that the healthy host does not suffer from the disease, but the host can remember this particular antigen, this particular pathogen, and then for a subsequent exposure the host can generate a quick and more efficient response, right? So we will also discuss different types of vaccines in this case. So vaccines can be of various types. So they can be generated by various processes, and according to that we can classify them in different types. So first is the live attenuated vaccine. So from the name you can understand that these live attenuated vaccines they lose their pathogenicity. So the attenuated means, they are attenuated, which means they cannot be pathogenic anymore. So they are not pathogenic, but they can transiently grow inside the host. So the transient growth occurs inside the host, which means that they can grow inside the host where we are whom we are vaccinating, but they cannot cause the disease. So can grow means it can replicate inside the host, right? So these attenuated vaccines can be naturally attenuated. For example, the Vaccinia virus. So Vaccinia virus, which causes the cowpox. One classic example is this cowpox virus. So this cowpox virus, it can, if it is inoculated in healthy humans, it confers immunity to smallpox, right? But it does not cause the disease. So it is a naturally attenuated live virus. But attenuation can also be achieved by growing the pathogenic bacteria or the virus for prolonged time under abnormal culture conditions. So for example, if we take this example of BCG, BCG is what? BCG is Bacillus calmette-guerin. This is a vaccine which is against the Mycobacterium bovis. So an attenuated strain of Mycobacterium bovis was developed. Mycobacterium bovis. So this attenuated strain of Mycobacterium bovis was developed by growing this particular pathogen in increasing concentrations of bile. So I told you that they are grown in abnormal culture conditions, so that after a long time they are severely attenuated, so that they cannot grow in normal conditions in normal host anymore, right? But they can be suitably used for vaccines. So attenuated vaccines, live attenuated vaccines, for example, you can see here the measles vaccine, mumps vaccine, the Sabin polio vaccine, rotavirus, rubella, these kind of vaccines are commonly used, which are live attenuated vaccines. And there are several advantages and disadvantages of a live attenuated vaccine. So the advantage is that since they are capable of growing inside the host, so they provide prolonged immune exposure to the host, right? So since they mimic the real growth pattern of the pathogen, right, not the attenuated pathogen, but that is why they have increased immunogenicity and they can efficiently produce memory cells, right? So the major advantage is this prolonged protection by the live attenuated vaccine. So that is why, since they provide prolonged immune exposure, so therefore it is some live attenuated vaccines require only one single immunization, and they do not require any booster shots or any booster doses, right? However, for example, and also one more thing, that is they can also since they can replicate within the host cells, so therefore they can induce this cell mediated immunity as well. Because they are growing inside the host cell, they can generate this cell mediated immune response as well. Now the oral polio vaccine which is the Sabin vaccine, this for example, this contains 3 attenuated strains of the poliovirus, 3 attenuated strains together of the poliovirus. But remember that this Sabin vaccine, it requires 3 shots at least, or like 2 booster doses it requires, because in one single shot the replication of the because there are 3 strains, so the 3 strains interfere with each other's replication, which is why it requires 3 shots, right? So this is the major advantages of a live attenuated vaccine, that it provides prolonged protection as well as cell mediated immunity it can induce. But remember, there is a major disadvantage of live attenuated vaccine, which is since they can replicate within the host like the natural real pathogen, so sometimes it may happen, and very very rarely it may happen, that this can mutate and revert back to the virulent forms, right? this is the disadvantage. Reversion to virulent form.
Surasree, this
Yes.
is like we knock out the gene, right? Like which is producing that particular
No, no.
pathogen of it, right?
There is no knockout or
So then
nothing is
here,
involved. The
what?
attenuation occurs by treating the pathogen or culturing the pathogen in under abnormal conditions, under abnormal growth conditions. For example, this BCG, the Mycobacterium bovis, is grown in increasing concentrations of bile. So, so bile is not a natural medium of growth for Mycobacterium bovis, and if we grow this M. bovis for long time in bile, so they will be attenuated so that they cannot infect any natural host, any healthy human being. It cannot infect anymore, and that strain
Okay.
will be used for vaccine.
So it may come at any point to its.
So that is the disadvantage, that it may anytime mutate to its virulent form. That is why. But it occurs very very rarely, and in case of this oral polio vaccine, which is the Sabin vaccine, it the rate of reversion is about 1 in only 2.4 million vaccine doses. Okay, so therefore the rate of reversion is very very low, but however this risk always remains with this live attenuated vaccine. Okay, now coming to the next type of vaccine, which is the inactivated or killed vaccine. Inactivated or killed vaccine, which is when the pathogen is killed. Okay, so there is a treatment involved which treats the pathogen with heat or chemicals. So heat treatment or chemical treatment is involved, which kills the pathogen and which makes it incapable of replication inside the host. So replication incompetent, because it is not live anymore. Okay, so it's treated with chemical or heat, which is why it is better than live attenuated pathogen, we can say, because it cannot cause the reversion any way. The reversion is not possible, so it's much more stable and it is safer than live vaccines. And also remember one thing, that since we are treating these pathogens with heat or chemical, so we have to remember that there are epitopes on this pathogen which are recognized by our immune system, and since we are using this as a vaccine, our immune system has to recognize these epitopes as in the real pathogen, and therefore our immune system will be primed for the subsequent exposure. So it is important that the epitope structures which are present on the surface of the pathogen have to be maintained, and we have to ensure that these structures remain okay for the immune system to recognize them, so that the heat or the chemical treatment cannot destroy these epitope structures. And one more thing is that the killed vaccines, the inactivated killed vaccines, they may require repeated boosters. Okay, so boosters shots are usually required, and because they do not replicate in the host, so therefore these killed vaccines do not also produce any cell mediated response, so they usually produce only humoral response. Right, so antibody mediated response is mainly produced by these heat inactivated or killed vaccines. Okay, so one good example is the polio vaccine of the Salk, right? Salk polio vaccine, and so it does not induce cell mediated immunity, or it also does not induce the secretory IgA response. So what happened is that however, there are some little disadvantages which are also present in this inactivated or killed vaccine, which is, so since it is killed or inactivated, it may generate weaker response than the live vaccines. And also we have to ensure that since we are killing or we are inactivating this particular pathogen, this inactivation has to be complete, has to be proper. Otherwise, this all the virus that we are actually injecting in the virus or any pathogen that we are injecting in the in a healthy individual, if all the pathogen are not killed properly, then they may cause the disease, which is not required. So therefore, this inactivation has to be proper and has to be complete. And so we can see that cholera vaccine, influenza vaccine, so the yearly flu shots, the flu shots that are given are basically your inactivated or killed vaccine, and also hepatitis A, polio, rabies, this kind of vaccines are inactivated or killed vaccine. All right. Any doubts so far? Okay, if not, we will move on to the next type of vaccine, which are usually purified.
Ma'am,
Yes.
ma'am, what is mean by Sabin vaccine and the salt vaccine? What is the difference between both, ma'am?
So the difference is that in the Sabin vaccine, the polio strains, the poliovirus strains were attenuated. So they were grown under abnormal culture conditions so that they could not grow in natural host anymore. But in case of the Salk vaccine, so the difference is that they were inactivated by formaldehyde, by the application of formaldehyde. So the formaldehyde killed the poliovirus and then they were used as vaccines. So when they are now injected into the healthy individual, the structure, the epitopes of the vaccines are still, of the virus is still intact, so that the immune system can recognize it and can produce immune responses accordingly. Okay?
Okay, ma'am. Thank you for your clearance.
Thank you. Okay, so now next moving on to another type of vaccines, which are basically purified macromolecules. In the previous section, we were discussing about the whole organism vaccines. Either they were live or they were killed. Now we are coming to the types of vaccines which are nothing but macromolecules, not the whole organism, right? So we will. So these are mainly called as subunit vaccines, and they are mainly of three different types. One can be a toxoid, and there can be the capsular polysaccharides, for example, or surface glycoproteins, which can be used as vaccines, and also recombinant vaccines. So coming to them one by one. First of all, coming to the toxoid. So which toxoids are generally produced in these diseases, diphtheria and tetanus. So what happens in diphtheria, I can explain. That is when so it is an infection with Corynebacterium diphtheri, a bacteria, and when it infects humans, what happens is that there is actually a virus which resides inside this bacteria. So this virus, which is called the fudge beta virus, this produces a toxin which is very potent in causing disease, and it destroys the underlying tissue and it causes damage to the heart, liver and kidney. And also what it does is it forms a fibrous tough layer in the respiratory tract, okay, which causes suffocation. So these kind of events occur in the diphtheria, and it is not caused by the principal bacterium, but it is caused by this particular toxin. So therefore, if the individual has to be immune against this particular disease, then there has to be an antitoxin which should be introduced in this particular organism or the individual, right? So that is a toxoid. So a toxoid is nothing but an antitoxin, and it is so what is done is that this particular exotoxin is purified and from the bacteria, and then it is inactivated with formaldehyde. Then it forms this toxoid, right? So it is inactivated by use of formaldehyde, and then vaccination with this toxoid induces this antitoxin antibodies, right? So which are capable of binding to the toxin and neutralizing the toxin. Okay, so antitoxin antibodies are produced which bind to the toxin and neutralize the toxin. However, you remember one thing, that this does not produce long-term immune response, and it only produces temporary protection. Okay, so this is a major disadvantage of the toxoids. Next, coming to the capsular polysaccharides, which is one very good example of this is this streptococcal pneumonia. So this is the pneumonia vaccine that I'm talking about. So what happens is that some pathogens, they have this hydrophilic polysaccharide capsule around them. So they have these polysaccharide capsules around them, which actually prevent phagocytosis of these pathogens, right? So they can evade phagocytosis. So therefore, if this particular capsule can be coated with antibodies and complement proteins, then that phagocytosis becomes easier. So this vaccine, this pneumonia vaccine that is currently in use for the Streptococcus pneumoniae, this vaccine contains 13 distinct capsular polysaccharides of this Streptococcus pneumoniae, right? So capsular polysaccharides of this Streptococcus pneumoniae are combined, and this vaccine induces formation of the opsonizing antibodies that can opsonize this particular organism. Now, another example is meningitis. So meningitis is caused by Neisseria meningitidis, and that vaccine also contains purified capsular polysaccharides, which can induce this immunity against the meningitis disease. Alright, so this is the capsular polysaccharide vaccines, one type of subunit vaccines. Next, coming to the recombinant vaccines. So the gene which encodes the immunogenic protein, it can be cloned and it can be expressed in cultured cells using recombinant DNA technology. So whatever gene is producing a particular the antigen can be cloned and expressed in any particular culture in cultured cells, right? And therefore, from this cultured cells, this particular pathogen can be purified, can be extracted and can be purified. So that is how the recombinant subunit vaccines are produced. So for example, this toxoids, the toxoids also can be the gene for actually producing this toxoid. The toxin, actually the gene for producing the toxin, can be cloned and can be purified from cultured host cells, and therefore these proteins can now be also used as vaccines, right? So that will be a protein vaccine. So one such recombinant antigen vaccine which is approved, the first recombinant antigen vaccine which is approved for human use is the hepatitis B vaccine. Is the hepatitis B vaccine, which is developed by cloning the gene for the major hepatitis B surface antigen, which is the surface antigen of hepatitis B in yeast cells. So this particular gene is cloned in yeast cells, and then these yeast cells are grown in large fermenters, and then this protein is purified in large quantity from these yeast cells. And so this particular recombinant surface antigen of the hepatitis B can now be introduced into healthy individuals as vaccines, and it induces the production of protective antibodies, and it's a very promising type of vaccine which is a protein vaccine, right? So in these types of vaccines we are discussing the subunit vaccines or the purified macromolecules, right, which is very different from the whole organisms that we were discussing previously. Now coming to the other experimental types of vaccines are the DNA vaccines and the recombinant vector vaccines. For example, let's talk about the DNA vaccines first. So this is a very recent vaccination strategy that has been developed, which utilizes plasmid DNA. Okay, it utilizes plasmid DNA which encodes these antigenic proteins. Encoding antigens. And then these plasmid DNAs are developed which encodes these antigenic proteins, right? And this plasmid DNA is directly injected into the muscle of the recipient. So this strategy relies on the host cells to take up the DNA. So therefore what will happen, the host cell takes up the DNA, the plasmid DNA, and then what happens is that it produces this protein. It produces this protein antigen. Now this antigen, therefore this antigen, since it is produced intracellularly, it can be presented with the help of MHC class 1, right, which can enhance or which can induce the cell mediated responses or cytotoxic T cell responses, right? So this DNA can either integrate into the chromosomal DNA or it can remain as an episome. And so this particular DNA vaccine might require some booster, some follow-up booster with some protein antigen, which is called a DNA prime and protein boost strategy. So first this DNA vaccine is inserted, and then the protein, then in the booster a protein is injected into the individual. Or CpG DNA, for example. CpG DNA can also be introduced into this plasmid, because you remember that CpG DNA is a ligand for TLR9, right? So it can also enhance the innate immune responses by this process. What are the advantages of this particular DNA vaccine is that since there is this encoded protein is in the host is expressed in its natural form, there is no modification, there is no inactivation. So therefore the immune system is directed against the antigen as exactly expressed by the pathogen, right? So there is no modification, like for example happened for inactivated vaccines or the toxoids, right? So there is no modification of the particular protein that is being expressed. And also a very important thing is that it can strongly induce humoral and cellular immune response. And since it is a plasmid DNA, it is very easy to store. It does not require very lower temperatures for storage, and it can be easily transported. So these are the several advantages which are there is for a DNA vaccine. Now some so an improved method of administering DNA vaccine is also that DNA vaccines are coated with gold microscopic particles, so gold nanoparticles, right? So the DNA vaccines are coated with gold nanoparticles for enhanced, like for better delivery of these DNA particles, or it can be also delivered by gene guns. So this causes rapid delivery of the vaccine. So that's all about DNA vaccines. And last we will talk about
But
the
but it will not change the our DNA script, or it may change like some one two protein in the DNA because it's hitting the nucleus.
so you are asking whether it will affect the host's DNA, own DNA.
Right.
No, that does not happen. Even if it is integrated into the chromosome, like this usually does not cause any major issues.
Okay.
Okay. Okay. Last, I'm going to talk about these recombinant vector vaccines. So recombinant vectors. So what is done is that so this particular so there is a gene which encodes a particular antigen, and this gene is inserted into a vector or a recombinant vector. So so it is actually individual genes that can encode these key antigens can be introduced into attenuated viruses or bacteria. So we are using many attenuated viruses, right? The attenuated, live attenuated viruses, which are used as vaccines and which is very standardized. Now, if we take a gene for any other pathogen, right, and then we insert this particular gene in any of these attenuated virus strains, then what happens or attenuated vaccines, then what happens is that, and then it is introduced into the host, then this attenuated organism serves as a vector, right? And therefore, since it replicates within the vaccinated host and it expresses the gene product of the pathogen, right? This particular part of the gene which we are administering inside the attenuated organism will be able to replicate inside the host, because this attenuated organism is able to replicate inside the host, and then it will encode the encode the particular antigen inside the host, right? So for example, this genetically engineered vaccinia virus is genetically engineered, which expresses high levels of the inserted gene product, and it can serve as a potential immunogen within a inoculated host. Okay, and one thing is that since this vaccinia virus, if you remember this experiment by Edward Jenner, so this vaccinia virus can also be introduced into the host by just scratching the skin, okay, or it can be introduced by causing a localized infection. So the foreign gene product which is expressed by the vaccinia virus, for example, if it is a viral envelope protein, then what happens, this then this envelope protein will be inserted into the membrane of the host, and therefore if it is inserted into the membrane, it can also induce cell mediated response, right? So not only humoral response, but also it can generate cell mediated response as well. One good example is this AstraZeneca vaccine. So this AstraZeneca COVID vaccine, or which is sold in India in the name of Covishield, it is a recombinant vector vaccine. So So you can see this diagram here, which shows that this COVID-19 virus, the spike protein of the COVID-19 virus, can be recognized in our body, and the body can generate immune responses against this spike protein. So what is done is that the genetic material which encodes this spike protein is incorporated in a vector, and then this viral vector is produced as a vaccine. Now what it does inside the human cell, when this vector enters into the human cell, it causes the expression of this particular spike protein, and it causes expression of this spike protein on the surface of the human cell, and then it can be recognized by the immune system, and it can produce, the immune system can produce antibodies against this particular spike protein. So this was the strategy for the AstraZeneca COVID vaccine, or the Covishield COVID vaccine, the recombinant vector vaccine.
So this is not the DNA vaccine. This does not use the DNA vaccine type.
Actually, it is very similar to DNA vaccines as well, because DNA vaccine is also a plasmid vector which is expressing the particular antigen. Here also it is a vector which is expressing this particular antigen.
But it is not going to the DNA, like it's not touching the DNA or going to the nucleus.
It is coming actually. So the point is that it will not coming into the nucleus. It will not come into the nucleus, because this particular vector, it will replicate inside the host, and what will happen is that once it replicates, it will also express this protein, and once this protein is expressed, it will be expressed on the cell surface.
Okay.
Am I clear?
But for DNA vaccine, first it should go like from DNA to RNA. All this translation has to happen, right?
This occurs in case of this recombinant vector vaccine. You are correct.
Oh, for this also it happens, but
exactly,
it does not happen in the nucleus.
No, so it is actually so this plasmid DNAs are not replicated inside the nucleus. They can be replicated outside, because they actually have this machinery to replicate their own
by themselves they do, like they don't touch any host cell organelle.
Yes, they can replicate, and they do not depend on the host's machinery for that.
Oh, like both DNA vaccine as well as is recombinant.
Yes, more or less, yes.
Oh.
Okay.
Yeah.
All right, and one other type of vaccine which was very commonly used during this COVID-19 pandemic, which was developed by this Pfizer, and another vaccine which was developed by Moderna, they were mRNA vaccines. Okay, not DNA vaccines, but mRNA vaccines. So what they did is that they took this mRNA for the spike protein, and this mRNA was given as a vaccine, and when this mRNA is entering into the host cell, then this mRNA is from the mRNA, the protein synthesis occurs, right? And then it similarly produces this COVID-19 virus spike protein, and then it is recognized by the immune system, and specific antibodies against this COVID-19 virus can be produced. So this is a strategy which was used by Moderna vaccine as well as the Pfizer vaccine.
I heard a lot of controversy about this vaccine. Like it stays and then it's always producing that spike protein, and we always like responding to the immune response. Like even like it should happen like one time and then once neutralized, this should go away, right?
Actually, the thing is, once our immune system recognizes this particular, what I think is, once this immune system recognizes this particular antigen, it will elicit an immune response and it will generate these antibodies. But I don't think that it will always elicit an immune response in the body. It's not. I
Like
don't think it's
if,
like
if
that.
it is copying like that mRNA is copying, because my understanding is this
No, this
mRNA
mRNA
going
is
to the cell,
no.
that cell
Actually, what
will be
happens is that
cycle.
mRNA has a very short shelf life. So mRNAs
Okay.
are quickly degraded in the cell. It's not like they are going to stay for a long time. So once
Also,
it
that
generates,
cell should be destroyed, right?
this protein, once it synthesizes the protein, after that this mRNA can be degraded. And if it is degraded, then of course this protein also has a short shelf life. So after that time, when the protein is degraded, then there will be no such immune response, no such prolonged immune response.
And also, like as they are expressing on the cell surface by
Yes.
MHC1, I believe. So then it will also be gobbled by macrophage or something, right? This
Yes,
cell,
yes,
particular cell.
yes,
Okay.
definitely.
And somehow, if it release in the blood, like let's say some error happened and this mRNA released in the blood, is there any implication that can happen on this?
Similarly, as I told you, like there will be immune responses which will be elicited, but that will not be prolonged because
Uh,
mRNA will be degraded after some point of time. So
and it does not have any capability to replicate itself, like
no,
mRNA.
no,
Okay. Okay.
unlike DNA vaccines.
Yeah.
Okay. So that was the mRNA vaccine, and another vaccine which was actually um, Novavax, for example, which was another COVID vaccine. It was a subunit vaccine. So this is the example of this subunit vaccine. So here what was occurring is that this spike protein was purified from animal cells or yeast cells, and this purified spike protein was used as a vaccine, right? So this is a typical example of a subunit vaccine. And then when this spike protein is introduced in the healthy individual, it can boost immune responses against the COVID-19 spike protein, right? So this was the strategy which was used for Novavax.
Like subunit means only that epitope we are transferring into the cell, is it?
Not the epitope, but the whole protein, the whole spike protein.
Oh, so then it will go
Subunit in
degrade
the
and
sense, why? Because it is not the whole organism. That is why it is a subunit.
oh, like one part of it.
One part of the
Like
whole
we're taking one.
organism. Yes, which can elicit the immune response. Not any part of the organism will go as a subunit.
Okay.
Which
So
is
then it will
something
taken up in the.
which is characteristic of that particular pathogen only, and not something which is very similar to the self, right? So otherwise the immune system will not be able to recognize that.
Like we have to process it in that way that
Exactly.
so that it will elicit the immune response.
Exactly. And
And
whenever there is
after
a
that,
subsequent exposure, the immune system can recognize that this was previously, I had exposure with this previously, so I need to activate my memory cells like that. So that whole network has to be working for that.
oh, and after that, I think it will chop also, right? Like to make
Of
that
course.
particular epitope.
Of course, of course.
obviously. Okay.
Okay. Alright, so these were about the different types of vaccines that we discussed. And one important concept that I want to give you here, which is herd immunity, something which became a very known common term in this after this pandemic. So vaccination, as we know, that it is not 100% effective, which means that even if we immunize the whole population with vaccines, there will be certain individuals, suppose the aged individuals or the very young individuals, the babies for example, for them the vaccines won't be 100% effective. So a small percentage of recipients will always respond poorly, and therefore they will not be adequately protected. But remember that if the majority of the population is immune to any infectious agent, then it significantly reduces the pathogen reservoir, which means that if, for example, there is a group of 100 people, in that if 95 individuals are after vaccination, they are immune to a particular pathogen, then what will happen? If they are immune, so they will not cause the disease, and if they do not cause the disease, these 95 people, if they do not cause the disease, they will prevent the spreading of this disease as well, right? So even if there is an infection they are facing, they will not cause any disease, there will not be any disease in their bodies, and they will not spread the disease as well. So therefore this phenomena is known as herd immunity, in which the chance of a susceptible individual containing contacting an infected individual is very low, right? If the other 5 people who are not very protected by the vaccine, if the other 5 people who are not very protected by the vaccine, if they do not get contact with the disease anyhow, then they will not be they will also not show any disease. So therefore they will also be immunized if the majority of the population is immunized. So that is known as herd immunity. Okay, so if the majority of the population is immunized, that will prevent the spreading of the disease, and that will actually help the poorly immunized individuals also to escape the disease, to avoid the disease. Okay, if there is no other issues, then we will move on to the last topic for today, which is the Hybridoma technology. So Hybridoma technology is the technology by which monoclonal antibodies can be produced in large quantities. So this technology was developed by Kohler and Milstein in 1975. So what they did is that they fused a normal antibody producing B cell with a cancerous myeloma cell. Why is this fusion taking place? So first of all, let me introduce these two terms, polyclonal antibody and monoclonal antibody. Monoclonal antibody simply means that these antibodies are directed against any one particular epitope, a single epitope of the antigen. So for example, here you can see that a particular antigen can contain many different epitopes, right? But monoclonal antibody is the antibody population which is directed against, or it can recognize against a single epitope of the antigen. However, polyclonal antibody is nothing but a population of antibodies. Okay, so a population of antibodies which can recognize various epitopes on the antigen. which can recognize various epitopes on the antigen. Right. So now, what is the need for producing monoclonal antibodies? So monoclonal antibodies are can be widely used. So they are now widely used for diagnostic purposes, for drug purposes. They are also fused with enzymes to so they are now called enzymes, which have varied uses nowadays. So monoclonal antibody, why do we need monoclonal antibody? Because you can see that in polyclonal antibodies, since there is a large population of antibodies which can recognize various epitopes on the antigen, there can be cross reactivity as well. To reduce that kind of cross reactivity, we use monoclonal antibodies. So monoclonal antibodies are much more specific than the polyclonal antibodies, right? Now monoclonal means they are generated from a single B cell clone, right? A single B cell which expresses only one particular type of antibody. Right. Now, how to generate these monoclonal antibodies? So what Kohler and Milstein did is that they isolated the normal spleen cells or the plasma cells from a mouse, which is immunized with this particular antigen, right? So the normal plasma cells, these normal plasma cells were isolated from this immunized mice, and these normal plasma cells were fused with these myeloma cells, right? Now there are some important concepts here. That is, these myeloma cells are HGPRT minus cells, which means they are mutant myeloma cells which lack this HGPRT enzyme. What is HGPRT? Hypoxanthine guanine phosphoribosyl transferase. This is a particular enzyme which is required for the nucleotide synthesis, okay? And it is an enzyme which is involved in one of the pathways of the nucleotide synthesis or DNA synthesis, which is the salvage pathway. So DNA synthesis occurs in two pathways. One is a salvage pathway, another is a de novo pathway. Okay, now HGPRT enzyme is involved in this salvage pathway. Okay, the alternative pathway, so since the myeloma cells are HGPRT minus, what I mean to say is that the myeloma cells do not produce this HGPRT enzyme, so therefore this salvage pathway of DNA synthesis will not be occurring in the myeloma cells. No salvage pathway, right? So therefore the myeloma cells have to use this de novo pathway for their DNA synthesis, right? So now first of all, what is happening is that these plasma cells are fused with these myeloma cells. How does this fusion take place? By using chemical fusion agents, for example polyethylene glycol. Polyethylene glycol or PEG is used for this kind of fusion. And once this fusion occurs, so always remember that it's not that only plasma cells will fuse with myeloma cells, because there are two different populations of plasma cell and myeloma cells. So the fusion can generate three outcomes. One is a fusion between a B cell and a B cell. Okay. Next is a fusion between myeloma cell and a myeloma cell. And the third one, which we require, is a fusion between a B cell and a myeloma cell. Why am I saying that we require this fusion of B cell and a myeloma cell? So a B cell can produce this antibodies against this particular antigen, right? And the myeloma cell is a plasma cell which can generate large amounts of antibodies. So if now these two cells are produced, what we can do, if now these two cells are fused, what it will happen is that this particular fused cell will produce large quantities of this antibody against this particular epitope of the particular antigen, right? So we need this fusion of B cell with a myeloma cell. Now, how can we prevent the fusion of a B cell with a B cell and a myeloma cell with a myeloma cell, which will not be required for our experiment, for our production of monoclonal antibodies? So for that selection, we need a selection medium, and this medium that is used here is the HAT medium. HAT medium is the selection medium. The HAT medium is nothing but hypoxanthine, aminoterine and thymidine. HAT stands for, H stands for hypoxanthine, A stands for aminoterine and T stands for thymidine. What are these? First of all, hypoxanthine is a purine derivative and thymidine is a pyrimidine, right? So hypoxanthine and thymidine will be used for DNA synthesis. So that is why they are provided in this medium. Okay, what is aminoterine? Aminoterine is one agent which inhibits the de novo pathway. Okay, aminoterine inhibits de novo DNA synthesis. So now let's see here what is happening. First of all, we are using a myeloma cell in which there is no HGPT enzyme. Therefore, this myeloma cell cannot synthesize DNA by salvage pathway. Next is, this myeloma cell cannot also synthesize DNA by de novo pathway because the aminoterine inhibitor is present in the HAT medium, right? So de novo pathway is also blocked, or de novo pathway is also inhibited. So therefore, the single myeloma cells or the fusion of the myeloma cell with the myeloma cell will not be able to sustain for long, or will be able to survive for long in that particular HAT medium, right? So therefore, we can cancel this out. This myeloma fusion, we can see that it will not survive for a long time because they do not have the HGPT enzyme. Also, the de novo pathway is blocked, right? So therefore, there will be no DNA synthesis. Therefore, this particular fusion will not be surviving for a long time. Next is the fusion of the B cell with the B cell. Now, both these two B cells are perfectly normal, so they are able to survive. But B cells in vitro, they have a very low shelf life, so they will naturally die after some time. They are not immortal cells, right? However, this particular fusion, the fusion of a B cell with a myeloma cell, will be immortal because this myeloma cell is a cancerous cell. It can divide continuously, and also because of this fusion of the B cell, they will produce a certain particular type of monoclonal antibody against the particular epitope of this antigen, and they will also produce large quantities of antibody which will be required for our future purposes, right? So this is the Hybridoma technology that was developed by Kohler and Milstein, and they also received the Nobel Prize in Physiology and Medicine for this development of the Hybridoma technology. Okay, any doubt from this? So once these fused B cell hybridoma, B cell myeloma cell clones are obtained, they are now diluted to single cell dilution so that the individual B cell myeloma cell fusions can be identified and monoclonal antibodies can be produced from that. Any questions so far? Okay, if not, then moving on to our question answer session. Please respond in the chat box. Yes. Okay, the correct answer is the myeloma cells which are deficient in this HGPRT enzyme. Next question. So take some time, match these carefully. This is the examples of the vaccines along with the types of the vaccines. Any other answers?
We have to remember all the vaccines and their virus correspondingly, is it?
Um, more or less, yes.
Okay.
So the correct answer here is, most of you guessed correct, A. So BCG, Bacillus calmette-guerin, is the live attenuated vaccine, whereas rabies is inactivated vaccine, and hepatitis B is recombinant vaccine. So option A is correct. Okay, moving on to the next question about inactivated vaccines. What are the correct statements about inactivated vaccines? Okay, yes, so the correct answer is B. So the solution is that the virulent pathogen is inactivated by chemicals, that is true. It induces humoral immunity, that is also true, and it cannot revert to virulent form because they are killed vaccines. And this is not right, that it requires a single booster dose. This is not correct because they require multiple boosters. And also it only stimulates mainly humoral immunity, so both cell mediated and humoral immunity cannot be induced. That is also wrong. Okay, moving on to the fourth question. Correct. So the answer is herd immunity. Okay, moving on to the last question for this session as well as for this whole run of the course, which is not a characteristic of passive immunization. Right. So the correct solution is C. Passive immunization does not give a long lasting protection. All right. So that's all. Thank you very much for joining. I wish you all the best for your exam. And if you have any doubt, you can post it in the forum. I will check and I'll try to answer as much as possible. And so this is the last session for this course run, and I hope you all the best. Um, so I hope you enjoyed these sessions with me. I quite enjoyed interacting with all of you. So that's all from my side. If you have any other doubts, you can always let me know.
Yeah, as always, I have doubt, but
Okay,
this
yes.
is this is a bit outside from what is that, immunology, but a little bit related to immunology. But before that, obviously, heartful thank you because you answer all my questions, all the sessions almost.
No problem. That is my duty only.
And
Thank you
is
all for
it like
actually interacting so much with me. I learned a lot during this process also.
And is it like we can connect after the course also? Like we
Yeah,
can get in touch.
yeah,
It's
sure,
okay,
no
right?
problem. My email ID is with all of you. It's in the forum also, from which I email. So you can always mail me. It's not a problem.
Okay, so
You can
you are
always get in
reachable.
touch. Yeah, sure. And thank you, all
Okay,
of
so
you.
yeah, so my doubt a little bit like autophagy has been discussed here.
Yes.
Okay, so uh, what happens that those proteins also express on the cell surface, right? Through MHC molecules.
Yes.
So then what happens to those things? Those does not generate any immune response.
No.
Then what happens to those things?
So that is recognized as self by the immune cells. So that is why they do not generate any immune response. But if you remember that after a particular protein is or an antigenic peptide is expressed by the MHC class 1 molecule on the cell surface, after some time it will be recycled. So what will happen, this MHC molecule along with the antigenic peptide will be taken in the cell once again, and it will be degraded by the proteasome.
Okay, so that is theoretical question. Now practical question is
Okay.
that I heard that like fasting as well as exercise both induce this autophagy.
There are actually several factors which can induce autophagy. So, yes, what is the specific question?
Question is that like then it induced autophagy, that I can read from many research paper or something, that fasting or exercise
Okay.
like easier way, but it needs to stay, right? Let's say if we are doing fasting, let's say after 48 hours it starts the autophagy, so it should stay for long, like if I'm not eating anything in that sense. But if I'm doing the exercise, yes, it induces the autophagy. But after that, let's say I'm eating something just after exercise and all, so that is stop the autophagy, something like that. Like that is my doubt.
Actually, so I am not very confident about this particular point, that is um, fasting and the relation of fasting and autophagy. But if you want a detailed explanation of autophagy, I have I have many cell biology books which I can share with you, and where you can get a more scientific and more detailed explanation of what happens in autophagy. So I think that will explain your doubt better. So if you want, I can
Okay.
share that.
Yeah. Sure. Yeah. No problem.
Okay.
Okay, that's all my doubt.
Okay.
Thank you.
Okay.
Thank you again.
Thank you, Shondeep. So, all of you, bye then. Take care. If you have any queries, you can always get in touch with me. And I hope you have a good exam. All right? Bye then.
Bye.
Ma'am,
Hello? Hello?